Hi and Assalamualaikum. My name is Fatina Nabila binti Khairuzamri. I am from Department of Chemical Engineering Technology or Faculty of Engineering Technology at University Tun Hussein Al Malaysia. In this video, I will present about my final year project entitled In Silico Structural and Functional Annotation of Selected Hypothetical Proteins from Escherichia coli strain IAI39. My supervisor for this study is T.S. Dr. Asha binti Muhammad Rehan, and my co-supervisor for this study is Dr. Eliza binti Muhammad. The ability of Escherichia coli to induce infections relies on its virulence factor machinery. There were 997 proteins of E. coli strain IAI39 extracted from NCBI database being hypothetical, which means that the function and structure of the proteins are yet to be characterized. Hence, by using in silico approach, this study aimed to annotate the function and structure of these unannotated proteins. Initially, all 997 proteins went through primary screening for pathogenicity. From there, 192 of them were found to be pathogenic and 10 of the pathogenic proteins were selected and underwent further physicochemical, functional, and structural categorization through an integrated bioinformatics approach by means of amino acid sequence and structure-based analysis. The obtained data revealed that 2 targeted proteins showed a high possibility to be involved in either cell viability or cell pathogenicity mechanism of the bacterium. Findings from this study can help in facilitating a better understanding of pathogenic ability of this microorganism and enhance drug development and target identification processes in the aim of improving Escherichia-related disease control. The problem statement for this study was that E. coli is the causative agent of several widely known diseases such as cholecystitis, bacteremia, colonitis, and urinary tract infection. To combat this bacterium, its virulence factors machinery, which mostly involves proteins and other essential components, must be fully studied and understood. However, 997 of 4,725 of E. coli strain IAI39 total proteins are still hypothetical and putative in terms of their structural and functional characteristics. The objectives of this study were to elucidate the physicochemical characteristics of selected hypothetical protein targets from Escherichia coli strain IAI39, to perform sequence comparison and sequence-based function prediction of selected hypothetical protein targets from Escherichia coli strain IAI39, to perform structure-based analysis of selected hypothetical protein targets from Escherichia coli strain IAI39, and to predict and annotate the structure and function of selected hypothetical protein targets from Escherichia coli strain IAI39. Escherichia coli, also known as E. coli, is a gram-negative, facultative anaerobic, root-shaped, coliform bacterium of the genus Escherichia that is commonly found in the lower intestine of humans. Most E. coli strains are harmless, but some serotypes can cause serious food poisoning in the host and are occasionally responsible for food contamination incidents that prompt product recalls. Escherichia coli includes not only commensal strains, but also pathogenic ones that cause a variety of human diseases, resulting in more than 2 million deaths each year. Strain IAI39, that was the focus of the study, is an extra-intestinal pathogenic E. coli strain, expect, isolated from the urine of a patient with urinary tract infection in France in the 1980s. As for expect E. coli, these pathotypes have many virulence-associated factors, including adhesins, toxins, ion acquisition factors, lipopolysaccharides, polysaccharide capsules, and invasins, which are usually encoded on pathogenicity islands, plasmids, and other mobile genetic elements. E. coli strain IAI39 belongs to phylogenetic group D and is serotype 07-K1. Interestingly, E. coli strains possessing the K1 capsular polysaccharide are predominant among cerebrospinal fluid sample isolated from cases of neonatal E. coli meningitis. Expect bacteria are transmitted via fecal-oral, household, sexual, and foodborne routes. Environmental E. coli that resemble the expect causing human extra-intestinal infections have been recovered from waterways, sewage, domestic and wild animals, soil and other environmental samples, suggesting multiple non-human reservoirs for human experts. In order to annotate the structure and function of Escherichia coli strain IAI39, in silico methodology was used. The first step in silico methodology was sequence retrieval. In sequence retrieval, the accession number and full sequence of each of the 997 conserved hypothetical proteins of Escherichia coli strain IAI39 were retrieved from National Center for Biotechnology Information (NCBI) website. The second step was physicochemical categorization. For physicochemical categorization, several chemical and physical parameters such as molecular weight, isolatory point, extinction coefficient, instability index, and gravity were analyzed using XPaSI ProgParam tool. The next step was variance prediction by MP3 server. MP3 server used support vector machines (SVM) or hidden Markov model (HMM) to calculate the algorithm and predict the pathogenesis of query protein. 393 hypothetical proteins were analyzed by this server for their virulence properties. The next step was conserved family and domain prediction. PFAM and NCBI CD search servers were used to predict possible domain or family of a query protein. Domain and family are able to give insight into the possible role or interaction that may be associated with the query protein by looking at the function and structure of proteins they are similar with. After that, subcellular localization was performed. PSOT-B server and Celo server was used to predict the subcellular localization of the query protein. This information was important in categorizing whether a protein is a membrane protein or a cytoplasmic protein. The next step was protein-protein interaction prediction by String server. String website is an online server that contains protein database of thousands of organisms and is useful in analyzing protein-protein interactions. By using String, the interactions between the query protein and other surrounding proteins were assessed. This enables the identification of functional and regulatory interactions among proteins. The next steps were secondary and tertiary structure prediction. Secondary structure prediction was performed by using psepreg server. The predictions allows the information on how many possible helices, strands, and loops are present in shaping the query protein. While for tertiary structure prediction, two different servers, namely Viral and XPaSI model, were used for each query protein. All two predicted structures were then validated using Ramachandran plot assessment and Q-min-4 score. From the validation, the best predicted structure was selected for structural refinement and further analysis. After that, structural refinement was performed. The selected three-dimensional structures with PDB format were subjected to structural refinement by Mode Refiner server for improvement. The visualization of the final structure was viewed using RCSB PDB 3D view. Finally, the last step was active site and ligand prediction. The refined tertiary model was sent to 3D ligand site for active site prediction and binding ligand. The server allows the prediction of the possible ligand binding sites and ligands of the query protein. As for results and discussions, the screening process of 997 conserved hypothetical proteins revealed that 192 of them are suitable to be the target proteins. 10 hypothetical proteins were selected as the target proteins in this study. In general, sequence and structure-based analysis showed that the targeted proteins are diverse in terms of their physicochemical characteristics, structures, and functions. Protein with accession number of CAR16995.1 showed small Q-min-4 score, which was -3.02, hence making it the best potential drug design candidates out of all 10 proteins. This was because Q-min-4 scores around 0 indicate good agreement between the model structure and experimental structures of similar size. The table shows the results for a protein with accession number of CR16995.1. As for conserved family or domain, the family revealed that this protein was classified into the family of outer membrane efflux protein, while NCBRC research revealed that this protein was classified into the family of multi-drug resistant outer membrane protein MDTQ. As for subcellular localization, both PSOT B and cellulose servers revealed that this protein to be at the outer membrane location. As for structural refinement, the favored region for Ramachandran plot assessment of this protein was predicted to be 97.48%, and the Q-4 score was negative 3.02. And as for active site and ligand prediction, zinc ion was predicted to be the binding ligand. The figure on the left showed the 3D structural refinement of the protein with accession number of CR16995.1, while the figure on the right side showed the structural view of active site and binding ligand of the protein with accession number of CR16995.1. As for conclusions, overall, it can be deduced that out of 997 conserved hypothetical proteins that had been screened, 10 of the proteins have potential to be further studied due to the predicted pathogenicity. In terms of functional and structural annotation, each protein that had been analyzed in the study involved directly or indirectly in the virulence mechanism of E. coli strain IAI39. The sequence and structure-based assessments showed that the protein with accession number of CAR16995.1 is the best candidate to be further studied. The information obtained from the study is significantly important for future works and researches such as molecular docking and downstream experimental handlings. These are some of the references that were used in this study.